In today's lecture, we're going to look at two different biomolecules, the proteins and the nucleic acids. This is part 1 of the lecture. In part 1, we will only discuss the proteins. At the end of this lecture, part 1 and part 2, we will understand what are proteins, the basic structures, functions, and different forms of proteins. We will also learn about a very important class of proteins known as the enzymes. In part 2 of the lecture, we will look at nucleic acids, the different types of nucleic acids, their structure and function. What are proteins? Proteins are the most complex organic compounds. They are composed of carbon, hydrogen, oxygen, nitrogen, and sulfur. If you recall, carbohydrates and lipids, they only contain carbon, hydrogen, and oxygen. Carbohydrates and lipids. Proteins, they have nitrogen and sulfur elements as well. Units of proteins are amino acids. What does this mean? Amino acids are the monomers of proteins, or you can call them the building blocks of proteins. Proteins have a wide range of functions and roles. They have structural roles or functions. They are found in cell membranes. They are found in our hair, nails, bacterial flagella, and also cilia. Proteins are catalysts. They are enzymatic catalysts. They speed up the rate of chemical reactions in the body. Without enzymes, life may not exist. Proteins also have a regulatory role. For example, hormones like insulin, they are proteins that have a regulatory role. They regulate the blood glucose levels in the body. Proteins are also involved in transportation. The membrane channels that control the potassium, sodium influx, these are all proteins in nature. Proteins can also be broken down to give energy. Proteins also have a defensive role. They act as antibodies. They are antibodies. And the complement system, this is another defense mechanism in our body, is also mainly made up of proteins. Let's look at a few examples of proteins. Proteins with structural role, keratin. This forms the hair, skin, and nails. Proteins involved in transport of oxygen in blood to cells. This is the hemoglobin in our blood. Enzymatic catalysts. Trypsin breaks down or hydrolyzes proteins. Sucrase hydrolyzes sucrose. Sucrose, as you know, as you remember, as if you recall, it is a disaccharide, carbohydrate. Proteins also have a storage function. Ferritin stores iron in tissues and releases it when the body needs it. Regulatory function, for example, insulin regulates glucose levels in blood. Contractile function, myosin, this is found in the thick filaments in the muscles. Antibodies, they help to fight infectious diseases. And sensory proteins such as the acetylcholine receptor, they aid in nerve impulse transmission. Amino acids, these are the monomers that make up the protein molecules. Essential and non-essential amino acids. Essential amino acids are those that are required for protein synthesis, but these cannot be completely synthesized by the human body. Some of the amino acids must be obtained from the diet. 8 essential amino acids that can be obtained from the diet are the tryptophan, lysine, methionine, phenylalanine, threonine, valine, leucine, and isoleucine. These essential amino acids can be found in meat, poultry, fish, eggs, and dairy products. Over here to your right, you see the structure of an amino acid. All amino acids have a hydrogen, a carboxyl group, and a amine group or amino group. The R group is different for different amino acids. Here we see the 20 amino acids that are required for protein synthesis. If you look carefully, these amino acids all are different because of their R group. They all have the same carboxyl group, amino group, and a hydrogen connected to the carbon in the center, and the R group is different for different amino acids. We have the non-polar amino acids. There are also polar uncharged amino acids, aromatic rings, like that, aromatic rings. We also have the positively charged amino acids. See, there's a positive charge here, and also the negatively charged amino acids. In a cell, there are more than 60 amino acids, but only 20 are used in protein synthesis. Now let's look at the structure of proteins. Proteins basically have 4 structures. The primary structure is actually the sequence of amino acids that are linked together by peptide bonds. So if you look at the diagram here, it is just a sequence of amino acids. So one amino acid is linked to another amino acid by a peptide bond. So this peptide bond holds the amino acids together. The secondary structure of amino acids has 2 forms. One is the alpha helix and the beta pleated sheet. Now what is different from the primary structure is that the secondary structure has got hydrogen bonds as well as the peptide bonds. The hydrogen bonds are holding the structure in place. And if you look at this alpha helix, what is holding this alpha helix together are the hydrogen bonds here between the hydrogen and the oxygen. The peptide bonds are between the amino acids here. Tertiary structure has got more bonds or more types of bonds. So the chemical bonds found in a tertiary structure include the hydrogen bonds, hydrophobic and hydrophilic interactions, peptide bonds that holds the amino acids together, disulfide bridges, as you can see here, disulfide bridges, ionic bond, and van der Waals interactions. The primary structure of the protein folds itself into complex dimensional structures which are held together by these chemical bonds, and this results in the formation of tertiary structure. Here is an example of a tertiary structure of a protein. This is ribonuclease. As you can see, it has one N-terminal, amino terminal, one carboxyl terminal. It also has a secondary structure, alpha helix, and another secondary structure, beta pleated sheet. Let's look at the quaternary structure of proteins. Quaternary structure is the result of 2 or more polypeptides linked by other bonds such as the disulfide bridges. For example, let's say you have a tertiary protein like that, and then let's change color. Let's say you have another tertiary protein, and these two are now linked together. Here, over here, linked together. Or it could be like that, linked together. So this is a tertiary structure, association of two or more polypeptide chains with each other. Example of a quaternary structure is your hemoglobin molecule. As you can see here, hemoglobin, it has got four different structures. One here, another green one, second purple, and this red one. All these structures, all these polypeptides, four different polypeptides are combined together to give you the hemoglobin molecule. So this is quaternary structure. This is a summary of the four different protein shaped structures. The primary structure, as you know, has only peptide bond. The secondary structure has peptide bond as well as hydrogen bonds. The polypeptide chain, the tertiary structure, has many bonds. For example, disulfide bridges, the ionic bonds, the van der Waals interactions, and so on. Quaternary structures, just like your tertiary structure, also has got many bonds holding the structures together. Denaturation of proteins. Denaturation of proteins involves the loss of secondary, tertiary, and quaternary structures. The proteins loses its shape and thereby loses its functionality. Denaturation of proteins can only destroy the bonds which are holding the secondary, tertiary, and quaternary structures. Denaturation reactions are not strong enough to destroy the peptide bonds that are holding the amino acids together. So when a protein is denatured, it returns to its primary structure. Okay, so if you have secondary or tertiary or quaternary structures, when they are denaturation, they return to their primary structure. So what can denature a protein? What are the factors that can contribute to denaturation of proteins? Extreme changes in pH, temperature, and salt concentration. These can destroy the bonds that are holding the secondary, tertiary, and quaternary structures together, and the protein returns to its primary structure. Now let's look at a special class of proteins known as the enzymes. Enzymes are catalysts. What do catalysts do? They increase the likelihood of reactions, chemical reactions taking place in the body. Enzymes speed up the rate of reaction. So organic catalysts of reactions which take place in the living organism are known as enzymes. Enzymes usually have names ending in ASE. For example, hydrolase is an enzyme, lactase, ligase, these are all enzymes. Enzymes are grouped based on their mode of action. So they are named based on what they do. For example, hydrolase depolymerizes macromolecules. Ligase, what does it do? It ligates. Ligates means to join. So it ligates two DNA molecules together. Oxidoreductase, it removes or adds electrons. So the names are based on their functions or their mode of action. Enzymes are very specific and will only work with certain substrates. They work based on a lock and key mechanism. So let's say if the substrate is the key, so only a particular substrate can fit into the active site of the enzyme. So here over here, you see, so this is the active site of the enzyme. This is the active site of the enzyme. So only a substrate that fits into this active site can bind to the enzyme. Lock and key mechanism. Enzymatic reactions are highly specific. An example is given here. The enzyme here is sucrase. How do we know this is sucrase? Because the substrate is sucrose. So the substrate sucrose binds to the enzyme, okay, forms an enzyme substrate complex, and then the enzyme breaks the sucrose into its constituent monosaccharides, the glucose and fructose. Enzymes can degrade a product. They can break down substrates into smaller products. For example, amylase can break down starch into simple sugars. Enzymes can also synthesize products. They can combine a fructose and glucose into sucrose. Factors that affect enzyme activity. A few slides ago, we talked about factors that can denature a protein. So if you remember, if you recall, factors that can denature a protein include extreme changes in temperature, pH, and salt concentrations. Since enzymes are proteins, these three factors also apply to factors that affect enzyme activity. Enzyme and substrate concentrations also affect enzyme activity. Presence of inhibitors also affect the enzyme rate of enzyme activity. Let's look at the effect of temperature on enzyme activity. So here the y-axis shows the reaction velocity and the x-axis shows the temperature. So when the temperature is low, let's say this is 0 degrees. Let's take this to be 0 degrees and let's take this optimum temperature here to be 37 degrees. So basically this could be an enzyme found in the human body. And here let's say the temperature is 70 degrees. Here is 0 degrees. At 0 degrees, we see that enzyme activity is very low because the enzyme is inactive. As the temperature increases, enzyme activity also increases up to a optimum temperature. The optimum temperature is when the rate of enzyme activity is at its greatest. The highest rate of enzyme activity takes place at the optimum temperature. If you increase the temperature beyond the optimum temperature, you see that the enzyme activity starts to decline. So this is the denaturation process. So finally, the enzyme becomes denatured. Extreme changes in temperature, especially when it is high above the optimum temperature, it can disrupt or destroy the bonds that are holding the enzyme together. So as you remember, the denaturation of proteins, when the bonds are destroyed, the protein, in this case the enzyme, returns to its primary structure. Now let's look at the pH of enzyme activity, the effect of pH on enzyme activity. Enzymes have large numbers of acidic and basic groups in its structure. So if there are extreme changes in pH, it can affect these groups. So when we talk about pH starts from 0 and all the way to 14. The neutral pH is around 7. So when you go towards 0, the pH becomes more and more acidic. If you go towards 14, the pH becomes more and more alkaline. The optimum pH is where the enzyme has its highest activity, and the optimum pH changes for different proteins based on their environment. For example, pepsin is an enzyme found in the stomach where the condition is highly acidic. It has optimum pH of 1.5. Trypsin is another enzyme, but this is found in the small intestine. Okay, it has a pH of around 7.5. So different enzymes have different optimum pH. Another factor that can affect enzyme activity are the inhibitors, the presence of inhibitors. An enzyme inhibitor is a molecule that binds to an enzyme and decreases its activity. There are two main types of inhibitors, competitive and non-competitive inhibitors. A competitive inhibitor binds to active site of the enzyme and prevents the substrate molecule from entering. Here you have an enzyme here, okay, and this is a substrate. So when the substrate binds to the enzyme, it gives you a product. But if the inhibitor binds to the active site first, it prevents the substrate from entering the active site. Thereby, no products are formed. So this is competitive inhibition because the substrate and the inhibitor are competing for the active site, the same active site. Non-competitive inhibition, inhibitor binds to any site, not necessarily the active site of an enzyme, and it alters the shape of the enzyme. It changes the shape of the enzyme. So when the shape of the enzyme is changed, the active site is also changed and it cannot recognize the substrate molecule, and therefore the reaction is slowed down or completely inhibited. So here we can see this, if you see this, the inhibitor binds to the enzyme but not at the active site. So this is the active site of the enzyme. So the inhibitor is not binding to the active site, it is binding to another place in the enzyme, but it changes the shape of the active site. See that? It changes the shape of the active site, preventing the substrate from binding to the active site. We have come to the end of part 1 of the lecture. In part 2, we will discuss the nucleic acids.